Hello. The title of my talk today is Gene Flow and Mating System as an Indicator to Monitor Genetic Changes over Time in European Beech and Silver Fir. I want to give you a short overview about my talk. I will start with a brief introduction to the concept of genetic monitoring, indicators and verifiers, and then about gene flow and mating systems. Then I want to explain a bit on the materials and methods, then move to the results of Abies, results of Fagus, and then compare seed lots between 2 years and compare saplings from 2 years. So all started in 1992 with the Convention on Biological Diversity, when they called for a concept to halt loss of biodiversity. The genetic monitoring came then a little bit after into place. But what is genetic monitoring? It's the assessment of a forest population's capacity to survive and reproduce under rapid environmental change. And this should be observed on a long-term scale. So in 1996, Nam Kung and al. Developed a system for foresters to manage the forest sustainably in terms of genetic diversity. And later on, they improved the system by reducing the measured parameters. But still, it was a very time-consuming and expensive and difficult to practically implement. And therefore, a German working group within a program for conservation of forest genetic resources and a pilot study developed easy to implement system. Also, experts from U4Gen working group on forest genetic monitoring worked on the simplification of a concept for genetic monitoring in forests. In general, forest genetic monitoring should answer several questions. One of them can be, are the levels of genetic diversity maintained over time within critical limits, or if populations are able to survive and reproduce. But also it is wise to observe if there are significant changes in the mating system over years, or if there is a change of genotypic frequencies over generations. Is there an effective population size change over time? Or if the fructification periodicity and intensity over years changes. So what I'm focusing in my talk is on the mating system and gene flow. And for example, one goal of a genetic monitoring program can be the conservation of the processes that maintain genetic variation. And an indicator for that objective could be gene flow, but it would not be a direct measure. For that, we need verifiers in the form of parameter data. And this is some statistics or some values that describe the indicator. Graudal et al. Stated in 2014 that there are so far no generally accepted indicators proposed to use on an international level. And here, briefly, you can see an overview of the numbers of indicators and verifiers that have been developed through the years. You can see various changes in numbers of indicators, numbers of verifiers. And that is where we started actually in the project LIFEGENMON. At first, we tried to have as much as possible indicators and verifiers to describe every possible change in genetic patterns. But then we came to the to the idea that we should have the most informative and most cost-effective verifiers and indicators. And therefore, in the end, we had we proposed 3 indicators and 22 verifiers. And for the indicator gene flow and mating system, we have 5 indicators. And this is also called the advanced forest genetic monitoring level, because it is compared to the basic, or to the basic level. It is very time consuming and expensive, but there is a great informativeness in this indicator. So what is gene flow and how it affects the mating system and the genetic diversity? Gene flow is the movement of genetic material of genes between populations of a species. So it is immigration or emigration. It can be through pollen. This is usually, if it is per wind, high gene flow. Through seeds, it might be a little lower, especially for heavy seeds. For winged seeds, as we see here, for Abies, it's a bit higher. So the gene flow is the movement of the genes. It can be beneficial, harmful, or neutral regarding the perspective. So in conservation genetics, maybe we don't want immigration of pollen from, for example, foreign material, or let's say material that is introduced from some other populations that might not be adapted to the local conditions. The other way around would be in tree breeding, that for the plus trees and the high value material for breeding, there would not be beneficial if there is any genes from outside coming into the seed orchard, for example. So gene flow is determined by the mating system, and this means the recombination and variety of the genes between the generations determines the level to which the genes are exchanged between individuals and populations. Climate change can affect gene flow and mating system patterns, and therefore severely also the reproductive success can decrease. Thereby the gene flow and mating system patterns impact on genetic diversity can be influenced. So we think that gene flow assessments should be part of genetic monitoring, because gene flow and and reproduction are at the beginning of every new forest generation, and that is also why it can be used as an early warning tool. If at the beginning of a forest the reproduction and the gene flow is not intact, then already at the youngest generation the genetic diversity can be low and thereby affect future generations. Random or non-random mating can affect gene exchange. For example, non-random mating can occur when there is no flower or few or less flowering synchronization, then only certain individuals can reproduce with each other. So this is called assortative mating if there are similar genotypes or phenotypes that mate, or it is called dissortative mating if different genotypes or phenotypes mate with each other. Some terms that we assessed in the course of calculation of some mating pattern and gene flow issues were, for example, the biparental inbreeding. This is the inbreeding either through biparental inbreeding when a plant mates with a related individual, but also the self-fertilization can cause inbreeding, and this is when a plant mates with itself. So biparental inbreeding can actually occur at various frequencies in many natural populations, and this can be calculated from genetic data. The same as a multilocus population outcrossing rate also is an estimate of the proportion of outcrossed progeny, and this include both the mating between relatives and also the unrelated individuals. An increase of the outcrossing rate might indicate the maintenance, if not an increase of genetic variation. And this will likely result in the maintenance of the population adaptive capacity. Then there is the effective number of pollen donors. It is the number of pollen donors contributing to each seed family. So if one mother tree produces seed, and these seeds are studied by genetic markers, then one can define the fathers that are contributing to this family. And if the higher the number, the higher is the effective number of pollen donors. And actually it can be much lower than the absolute number of contributing pollen donors. Yeah, as I said, the probability that progeny share the same father can reveal the extent of diversity in the pollen donor pool, and therefore it is an indicator of the effective number of pollen donors. Then there is the effective population size, which is the number of trees contributing the genes to the next generation. If the population is small, or even the effective population size is small, this will result in a high proportion of siblings. And the smaller the NA is, the probability is higher that two individuals come from the same cohort of a population, which are siblings then, sharing the same father or mother or both. So what did we actually do in our study? Maybe you have seen this already. This map is the countries participating in the project LIFEGENMON. And here you can see the forest genetic monitoring plots in Greece and in Slovenia and in Bavaria. And so each country had 2 plots, each for each species. Altogether, in each plot, 250 adult trees were sampled, 20 seeds per 20 mother trees. Saplings were sampled in 2 years, 200 in the year 2015 and 200 in the year 2019. And the genetic analysis were performed using 11 microsatellite loci. Statistical analysis were performed using MLTR, colony, and some R package diversity. Sorry. So first to the results for Abies. You can see here the numbers for the inbreeding of maternal parents. If they are around 0, then it's actually no inbreeding in the mothers of the seeds. So these analyses are done on the seed lots. Here you can see 20 seed families. And this number gives the inbreeding of the mothers of these 20 seeds each. So in Greece, we can see a little bit higher numbers than for Slovenia and Germany. For the biparental inbreeding, so the mating among relatives, also in Greece, we found higher numbers. Whereas for the multilocus population outcrossing rate, the values were also highest in Greece. In Germany, they were okay. In Slovenia, they were a little bit lower. And compared to the effective population size, we can see that in Abies, in German plot, we had the medium values. In Greece, we had the highest, and in Slovenia, we had intermediate values. If we go to the inbreeding itself, the German different generations, so this is the adults, the natural regeneration and the seeds, we have an increase of inbreeding. In Greece, it stays more or less the same. And in Slovenia, we also have a vast increase in the seed lot. Then we calculated relative migration networks. This is based on the NM on the left side and the DJost on the right side. And you see the connectedness of the generations. And also with this very shadowy lines, it is also, you can see the connectedness between the populations. But what you can see is here that for the Germans, this is very close, the connectedness of the generations. In Slovenia, it's medium, and in Greece, there is the least connectedness compared to the other two countries. The same goes for the other method that has been used. Also here for Greece is it the weakest. For Germany, it's the strongest. So if we look at the dominant pollinators in 2015, we can see that there is some trees, some fathers that are very abundant in Greece, but also in Germany and in Slovenia. It's more or less even, so to say that some trees are always have the most contribution, and then there is a lot of trees that have very few contribution. And compared in the numbers, the total numbers of pollinators is lowest in Germany and highest in Greece. The number of known pollinators was highest in Slovenia, and it was very low compared to this value in Slovenia for Greece. And here we can see the number of seed pollinated by known and unknown pollinators. And this was actually the known pollinators were highest in Slovenia, and the unknown, for that the unknown were the lowest. So now I come, to the results for beech. Here again, if you remember the slide from before for Abies, again in Greece the values for the inbreeding of the mothers are highest. The biparental inbreeding also is quite high, but also the population outcrossing rate is quite high. But compared to Abies, the Fagus effective population size is quite much lower than in Abies. Inbreeding as such was a bit different in the generations in Germany. This case from Greece is that in the seed is the lowest inbreeding, the adults have the highest, and in the Slovenia and plot, the different generation have more or less the same inbreeding rate. The relative migration networks are a bit different. So the German has in Fagus a bit less connectedness. The Slovenian have the highest, and the Greece have again the lowest. Sorry. Coming to the dominant pollinators, again the curve flattens here with more or less the same shape. Total number of pollinators were 92 in Germany and 120 again in Greece, the highest numbers. And again, the number of known pollinators were the lowest. And here again, you can see this time the number of seed pollinated by the known pollinators was the highest in Germany. So now I want to show you some results for comparison of two seed lots, one harvested in 2006 and one in 2016. We used the same seed trees. Seeds were collected by clambing, 20 seeds per seed tree and genotyped on 16 NSSR loci. And if you compare the genetic diversity values, you cannot see very much difference. Also with the heterozygosity, it was much the same. The only thing what was a bit different was the number of private alleles. So each of the seed lots had their own private alleles. The mating system showed in the inbreeding of the mothers, they showed no inbreeding. For the biparental inbreeding, so the inbreeding among the breeding among relatives was lower in 2016, but not that much than in 2006. The correlation of paternity is calculated here also. So this is the proportion of full SIBs that were detected. It stayed pretty much the same. Then we have the effective number of pollen donors. This means that from the 20 seeds per family, 18.9 were from different fathers, and here each seed even was from a different father. So there was a lot of pollen flow and reproduction, a lot of pollen from different fathers in the pollen cloud. Considering the effective population size, this was actually going a bit down in 2016 compared to 2006, but it was not dramatically. So something also to consider, again, the dominant pollinators. Here you can see in 2006, we found that the numbers here, you can see these were the fathers. The red trees were taken out or fallen down, so we cannot see them in the 2016 seed lot. But also the most dominant father, which we found here, is not present in the first, let's say, 30 most dominant fathers. But these numbers of the total the total number of pollinators didn't change much. Also the number of the known pollinators, so there was not so much difference in pollen from outside. But only 51 pollinators were the same in both years. So now to the natural regeneration compared between the years. Here we can also see again the dominant pollinators. There were some that were not from within the plot. These with the numbers are from within the plot in 15. They were a bit less dominant in the year 2009. And here are the numbers. So it's the total numbers of pollinators was reduced by 11. The number of known and unknown pollinators were also going down, going up and going down here of the unknown. And the number of saplings pollinated by the known pollinators was going up. And again, the same pollinators were only 13 in both years. If we consider the effective population size, this was going down from 215 to 219. For beech, we can see here that for the seedlings, we had some of the known fathers here. They do not occur in the first range here. So the number of known pollinators go down a bit also here, but not really. Again, a very low number of pollinators which are the same in both years. And actually the effective number of the effective population size goes up from 215 to 19. So here I want to sum up with the results. We found outcrossing rates that vary widely among populations in the two different species and between the countries. We found a lower outcrossing rate in a fur population from Slovenia, which indicates that part of the progeny is derived from outcrossing and part from inbreeding. We found high outcrossing rates in all three beech populations, which indicate outcrossing as a dominant type of mating. Then we found a higher level of parental and biparental inbreeding in Greece for both species, which we don't really know why. The conclusions for genetic monitoring can be that the observed slight decrease in effective population size can result in a higher proportion of SIPs in future generations. So that is why we should monitor and follow this development closely in the future. The first results of mating system now gives us a baseline data for long-term genetic monitoring, that it is important to continue with our observations because the mating system affects very clearly the genetic structure and adaptation of a population, and therefore it is important in order to understand forest population capacity so that it can cope with the challenges caused by future climate change. And now I want to thank all the collaborators for the great 6 years that we had together, because our project is finishing soon, and it was great fun to work with you, and goodbye.